Hello everyone, this lesson is going to be on buffers. In Science 30, one of the big topics is acid deposition, and we'll see how this topic of buffers is related to acid deposition. So buffers, what they are is they are compounds that resist changes in pH. So if you do have a solution, and we'll just say that the pH is 6, if you add an acid to the solution, then what we would expect is that the pH is going to drop, and it's going to drop fairly fast. However, if there is a buffer that's part of that solution that has the pH of 6, then we're not going to see nearly as fast as a drop. So yes, buffers are going to resist changes to the pH, and that's not just making the solution more acidic, it's also making it more basic as well. So how buffers do this, remember that for our definition of acids, there needs to be the donation of protons. So what buffers are going to do is when those protons are being donated, they're simply going to grab onto those protons. And what that's going to do is it's going to prevent the formation of hydronium ions. And because of that, the pH is simply not going to drop. Buffers might also grab onto the hydroxide ions, and that would prevent the pH from increasing. The greater the ability to capture the protons and prevent the formation of those hydronium ions, then the less the pH is going to change, or at least it's not going to change quite as rapidly. And that means that the greater the buffering capacity is going to be. And again, this is going to be connected to acid deposition. So limestone and sandstone, which is what we do have in the sedimentary rock that makes up, the bedrock in southern Alberta, and the soil coming from that bedrock as well is going to be derived from the underlying bedrock, the limestone and the sandstone. So they are less sensitive, to acid deposition, and the reason for this is they do have a very high buffering capacity compared to other parts of the country, and even in the very northeastern region of Alberta, which is made up of the Canadian Shield. So these ones here, they do have bedrocks and soils that are much, much more sensitive to acid and bases. They have fewer buffers. And so if there is acid deposition, then what we would see is that the pH would change much, much more rapidly in places like the Canadian Shield, Eastern Canada, than what we do find in southern Alberta. This is just a picture that is showing what a buffer is going to be doing. So in this picture at the upper left-hand side, what we have here is a strong acid, hydrochloric acid. So if you do add a strong acid, especially to a solution, that's going to drop the pH fairly quickly. So in this case here, what we do have though, and it looks like this is our representation of the buffer that it's showing in this picture. So when we do add the hydroxide to the solution, what we're going to have is the buffer that's going to grab onto those protons that would be donated. If there is the hydronium ions that are formed, again, that's what's going to drop the pH of the solution. So once we add the hydrochloric acid to our solution that we do have here, what we want to prevent, if we do want to have a buffer, is the formation of these. So that is the hydronium ion. So the buffer is going to grab onto those protons that are being donated by the hydrochloric acid and prevent the formation of these. Eventually, it will reach a point where the buffer is no longer going to be able to grab any more of those protons, and then eventually we will see more of those hydroniums that are going to be forming, and eventually we will see a drop in the pH if enough of that acid is going to be added. We saw a graph similar to this before, so we'll kind of focus on the acid here, again, because Science 30 does deal with acid deposition. So the graph that we saw previous to this was on titrations, and in that case, what we saw was that there was a very rapid change in the pH, and the curve looked like this. So very, very rapidly, we can change the pH of the solution just by adding a little bit of an acid. So with this graph that we see here, notice that in the middle, it's completely flattened out. And what this shows is now you need to add a lot more acid. A lot more acid needs to be added in order to change the pH. Eventually, again, we do reach a point here where that solution can no longer withstand any further changes in the pH, and it will then drop considerably. But in the middle, what we see is a flattening of the curve, and that is what the buffer is doing then, is it's preventing any changes in the pH. Kind of difficult to make out the details that it is showing on this map, but we can still see from the colors, and the title that it has here, The Potential of Soils and Bedrock to Resist or Reduce the Acidity. So if we have acid deposition, what it's showing here in green is that the soils here, which again, this would be in southern Alberta, Saskatchewan, Manitoba, those soils have a high ability to reduce the acidity and prevent changes in pH. Whereas if we go over into the Canadian Shield and the northeastern part of the province, everything that we see in red here, so the eastern part of Canada, pretty much all of Quebec, southern Ontario, doesn't have nearly that ability to resist changes in pH. It just doesn't have the high buffering capacity in the soil that we do have in, well, essentially the southern prairie provinces. You also have buffers in your body. Your normal body pH is fairly neutral. It's around about pH 7.4. So we do have buffers in your blood as well that are going to, again, prevent changes in pH. You don't want the pH going up. That's a problem. You don't want the pH going down. That's a problem as well. So you do have to maintain what we call homeostasis in terms of the blood's acidity, and it should be relatively neutral. So buffers that are present in the blood, they're going to remove the protons that would result in the acidification of the blood until those protons can eventually be eliminated by the kidneys. And in fact, that is one of the major functions of the kidneys, is regulating the body's pH by holding on to bicarbonate ions, which are going to increase the pH, and by getting rid of protons, which are going to decrease the pH. So through cellular respiration, what is going on in your cells is they're using oxygen, they're using fuel, and they are producing carbon dioxide. So carbon dioxide is going to be increasing in your blood, because that is where the carbon dioxide goes, is from your cells into your blood, eventually to your lungs, so you can exhale that carbon dioxide. But in the meantime, when this carbon dioxide gets into your blood, there's water. Most of your body, most of your blood is water, and it's going to react with the water, and it's going to form carbonic acid, which is a weak acid. And that's what it shows at the picture at the bottom here. So this is the carbon dioxide gas, which is dissolving in the water, and it's going to form this weak acid, which is the carbonic acid. So because this is an acid, what it is going to do is it's going to donate protons. So one of these protons is going to be stripped off, and then this proton here is going to be donated to water, and that's going to form hydronium, and that's going to result in a drop in the pH. So your body, of course, doesn't want that to happen. So what we do have is this bicarbonate that's going to grab onto those protons. So all of this is in equilibrium. So we do have carbon dioxide dissolving in the water, forming the carbonic acid, and that's also going to be in equilibrium with the formation of the protons, the hydronium, and the conjugate base, the bicarbonate. So this balance that we do have here in these equations that can go in both directions, this is referred to as the carbonic acid bicarbonate buffering system, and that just ensures that the pH doesn't go too low, but it also doesn't go too high. Again, you don't want either of these. You do want to maintain homeostasis, and through this buffering system, it regulates the number of protons and ensures that the number of protons present are going to keep the pH around this pH of 7.4, which is ideal in most circumstances. So back to the environment and dealing with acid deposition. So if there is acid deposition, there is a little bit of a problem with what is called leaching, and a couple of different kinds of leaching that I'll talk about here. There are useful ions that are in the soil that are useful for plants. The plants do need to take up, that plants do use for their normal cellular processes. So what leaching can do is it can remove these useful ions out of the rock and out of the soil, and that means that it's not going to be available to the plants. So when it is removed out of the rock, out of the soil, it's dissolving in the water, and this is then what is called leaching if it's being removed. And acid deposition, what it's going to do is increase this amount of leaching and the loss of the useful nutrients from the soil. So yes, it's the beneficial ones that we're talking about here, beneficial ones that are going to be essential for plants in order for their normal growth and their normal cellular activities. So acidification resulting from acid deposition, whether it's rain or snow or dry deposition, this acidification of the soil is going to decrease the ability of plants to take those nutrients out of the soil because they're simply being removed or lost from the soil. So let's take a look at a picture here. So the left-hand side is what normally should be happening. So precipitation is taking place. That's what these arrows at the top is showing. Eventually that water is going to percolate down into the soil here. And then from the soil, that is where these nutrients are taken up through the roots, up through the stem or the trunk, and eventually to the leaves as well, to all of the cells, tissues that we do find inside of that plant. So this is the normal process. There will always be some of these, and these are going to be some of the useful ions. So the calcium, the potassium, the magnesium, those are going to be useful ones, but some of them inevitably is going to end up in the groundwater, is going to end up in the streams, and then would not be available to at least the terrestrial plants in order to take it up. It would then be available to aquatic plants. If we do have acid deposition though, so what is the impact of this acid deposition? So when we are lowering the pH by adding protons to the water, this acid rain is going to remove the amount of nutrients that are going to be available to the plant. So what does that look like? Well, if we follow this down to the soil here, where we have the acidification of the soil, it's going to dissolve these nutrients out of the soil into the rock. So if we take a look at the arrows here, what we see is that many, many more nutrients are being removed from the soil. So that is the leaching that we're talking about. So again, you need to take a look at the size of the arrows. If we don't have acid deposition, we have less nutrients that are being lost from the soil. With acid deposition, we have more nutrients that are being lost from the soil. So if we take a look at the other arrows that are up here, without acid deposition, more is available to the plants. With acid deposition, take a look at the size of those arrows, the bands. Less of these nutrients are now going to be available to the tree and to the terrestrial plants themselves. So that's one form of leaching. There's another form of leaching as well. There are other elements that you do find in the rock in particular, but also in the soil, that can, rather than being beneficial to plants and living organisms, they can actually be harmful. So these are usually what we refer to as heavy metals. Heavy metals that are kind of locked up in the rock, and they're typically not moving around. They're not making their way into the waterway. So if we do have this form of leaching, then what's going to happen is these heavy metals, and I'll give you some examples here. So aluminum, cadmium, lead, mercury. These ones, they do have a negative effect on plant growth. And one of those negative effects I have here is that it reduces their ability to resist pests. So they can't fight off these infections quite as well if they do have these heavy metals that are taken into their cells and into their tissues. So with acid deposition, what's going to happen is more of these are going to end up in the water, more of it is going to end up in the soil, and more of it can be taken up by the plants as well. And this is going to have a negative effect on the growth of the plants, a direct effect on the growth of the plants. But there's more than that as well, through something that is referred to as either biomagnification or bioamplification. So in this case, the heavy metals, like the mercury, they can be converted into a form where they have organic compounds associated with it. So in this case, a methyl group, and now it's referred to as methyl mercury. So mercury on its own isn't necessarily harmful for us, but methyl mercury, if we take that into our body, that does cause some significant problems. It is toxic, not only to us, but to other animals as well. It's stored in the fatty tissue of animals, and it's not excreted as other toxins might be excreted. So what also happens is it's going to reach higher concentrations, and this is where the biomagnification or bioamplification comes in. It's going to reach higher concentrations in animals that are higher up in the food chain. So maybe not as big of a deal for the plants, maybe not as big of a deal for the herbivores that are directly eating the plants, but the carnivores, and especially the top carnivores, and that includes us, this is what it's going to have the most dramatic impact on. So what sort of things on us does it really have an impact on? The functioning of our nervous system, the proper functioning of the nervous system, and if a female is pregnant, on embryonic and fetal development, it can also have an impact on that. So let me show you a few pictures here. So this is showing the mercury, or rather the methyl mercury amplification that is taking place in this food web here. So we're starting with the base of the food chain. So these would be the photosynthetic aquatic organisms. And as they are consumed by these small microscopic organisms, if we take a look at the concentration of the, let's focus on the methyl mercury, very, very low concentrations. ppb is parts per billion. So very, very low at the base of the food chain in the producers. When we go up to the next levels in the food chain though, and when we take a look at the concentrations, we can see that now the numbers are starting to go up. So still not that terribly significant at the second level in the food chain here, but as we go higher up, these numbers get bigger, and they get, in fact, very, very huge eventually in the case of this top predator here, which is going to be the bass. And of course, as humans, we might be catching this bass, and it might be consumed by us as well. So this certainly isn't the case with all fish, and I don't want to give the impression that all fish are going to be toxic in terms of mercury and other pollutants, but some of them are, and especially the ones that are at the top of the food chain. So at this level, it would be toxic and it would be harmful for the bass in terms of the functioning of their nervous system, and possibly on the embryonic and fetal development of fish as well. And that, of course, would also end up being the impact when it is passed on to the humans. So very similar picture that I'm showing here, multiple different sources of mercury in the environment. So here it is showing natural sources like volcanic eruptions, human sources like burning coal in an electrical generating station that might contain that mercury coming from a mine, converted into methyl mercury in the environment. And then again, it's just showing moving up through the food chain here. At the base of the food chain, so eating organisms like these, probably not as much of a problem because the level of the methyl mercury is not that high. So this picture is actually showing us the amount of these that we should be consuming in our diet. And what the picture is kind of showing is that, yeah, you know, these are okay. We really don't need to be concerned about the amount of salmon and pollock and oysters that we're consuming, at least in terms of the methyl mercury, and most of these are considered to be very healthy. If we take a look at tuna, and it does vary depending upon the source, so here they're saying that canned tuna that you would take in through your diet, so you can eat this a few times a week and it's still not going to be a problem. So other animals that are at the top of the food chain though, so in this case the fish that we're talking about, so sharks, pike they have here, other kinds of tuna like albacore tuna, halibut, they are at the top of the food chain, so the top carnivores. So what they're saying is with these ones here, just be a little bit more careful in terms of the amount that you are consuming, and maybe these ones eating only a few times per month, and especially if you are a female that is pregnant, if you have a child that's growing inside of your womb, an embryo and a fetus, then it's maybe not such a good idea to be consuming those. And just one final picture here. So this one isn't for the heavy metals, but this is showing bioamplification or magnification for an organic compound. So this one here, DDT, so it is a pesticide. So this is a human-produced organic compound. And this is another one that we do refer to as persistent, which means it sticks around for a long time. So that can be good if we're trying to get rid of a particular pest, but it can be bad if it biomagnifies in the food chain, and that's exactly what this does. So here they're showing relative concentrations with these numbers, and what they're saying is that as we're going up the food chain, what we're getting is higher and higher numbers. So in the producers, maybe this level of 1,000, whatever the units are, maybe that's fine, maybe that's not a problem. Maybe it's not a problem for the zooplankton, maybe it's even fine for the small fish, but once it gets up to the larger fish, and especially in the top predators here, that's where it's going to become a problem because the concentration is so high. So this might have an impact on the survivability and the reproduction of those top predators.